Welcome to Developmental Biology. Today, we are going to explore one of the most fascinating topics in modern science, embryonic stem cells. These microscopic entities are the master builders of the human body, holding the potential to become any tissue we need. We will uncover what they are, where they come from, and even dive into the mathematics of how they multiply in a laboratory. Let us begin our journey into the building blocks of life. To understand embryonic stem cells, imagine you have a magical block of clay that can be sculpted into absolutely anything. In the human body, these cells act exactly like this magical clay. Before a developing embryo decides which cells will become the beating heart, the thinking brain, or the protective skin, it starts with a pool of these blank slate cells. They hold the ultimate biological wild card. Understanding them is like discovering the fundamental building blocks of life itself, giving us a window into how a single fertilized egg blossoms into an incredibly complex human being. Now that we understand their potential, let us look at where these cells actually come from. Formally, embryonic stem cells are defined as pluripotent stem cells derived from the inner cell mass of a blastocyst. But what does that mean? A blastocyst is a very early-stage embryo, essentially a microscopic hollow ball of cells that forms just a few days after fertilization. Inside this hollow ball sits a small, tightly packed cluster of cells called the inner cell mass. This specific cluster is the exact tissue that will eventually grow into the entire fetus. This diagram we are looking at perfectly illustrates the origin of these cells. On the left, you can see the blastocyst, which looks like a hollow sphere. The outer ring of cells is called the trophoblast, which helps form the placenta. But notice that dense blue cluster inside? That is the inner cell mass. The arrow shows how scientists extract this specific cluster and transfer it into a petri dish, shown on the right. Once in the dish, these cells are cultured and become the pluripotent embryonic stem cells used in groundbreaking research. Having seen where they come from, we must understand their defining characteristic: pluripotency. The word pluripotent is the most crucial part of their definition. Pluri means many, and potent means having the power or ability. Therefore, pluripotent cells have the power to differentiate, or specialize, into any cell type belonging to the three primary germ layers. These layers are the ectoderm, which becomes skin and nerves, the mesoderm, which forms muscle and blood, and the endoderm, which develops into the lungs and gut. They truly are the ancestors of every tissue in your body. This incredible ability to become any cell leads us to an important question. Why do embryonic stem cells matter so much to doctors? Their importance lies in their unparalleled potential for regenerative medicine. If a person suffers from a condition where specific cells die, such as insulin-producing cells in type 1 diabetes, or dopamine-producing neurons in Parkinson's disease, these cells offer a way to grow brand new, healthy replacements. Furthermore, they are incredibly valuable for testing new drugs safely on living human tissue in a petri dish, rather than risking an experimental medication on a human patient. To actually use these cells in medicine, scientists face a logistical challenge. They need millions or even billions of them, which means they must grow and divide them in a laboratory. Stem cells divide by a process called mitosis, where one mother cell splits into two identical daughter cells. Because each new cell can also divide, the population does not just grow steadily, it grows exponentially over time, much like compound interest growing in a bank account. To manage this rapid biological growth, scientists rely on mathematics to predict exactly how many cells they will have. This biological reality brings us to the mathematical model that makes stem cell research possible. The formula is n of t equals n0 times e to the power of r times t. Let us break down every piece. N of t represents the total number of stem cells present at a specific time. N0 stands for the initial number of cells we started with. The constant e is Euler's number, representing natural continuous growth. The variable r is the growth rate constant, which depends on how fast the cells divide. And t is the elapsed time. If the growth rate increases, the population explodes much faster. Before we move on, let us see this formula in action with a concrete example. Suppose a researcher starts with an initial population of 10,000 embryonic stem cells. The continuous growth rate is 0.03 per hour, and she needs to know the population after exactly 48 hours. We substitute our known values into the equation. Multiplying the rate and time gives an exponent of 1.44. Evaluating e to the power of 1.44 gives a growth multiplier of approximately 4.2207. Multiplying this by our initial 10,000 cells tells us she will have about 42,207 cells ready for her experiment. Now that we have used this formula to find a future population, let us tackle a more complex, real-world medical problem. Imagine a clinical trial requires exactly 1 million specialized cells to treat a patient. The lab currently has 50,000 cells, growing at a rate of 0.05 per hour. We need to prove exactly how many hours it will take. We start by plugging our target, initial population, and rate into the formula. Our goal is to isolate the time variable, t. The first logical step is to divide both sides by 50,000. This simplifies the equation to 20 equals e to the power of 0.05 times t, meaning the cells must multiply 20 times over. Continuing our derivation, we are left with the equation 20 equals e to the power of 0.05 times t. To bring the variable t down from the exponent, we must use a mathematical tool called the natural logarithm. By taking the natural logarithm of both sides, we can unlock that exponent. Because the natural logarithm is the exact mathematical inverse of the exponential function e, they cancel each other out on the right side. This brilliant algebraic move leaves us with a much simpler equation. The natural logarithm of 20 equals 0.05 times t. We are now at the final stages of our derivation. We evaluate the natural logarithm of 20, which is approximately 2.9957. Now we have a simple linear equation. To find the final time t, we just need to divide this value by our growth rate of 0.05. The calculation gives us a time of approximately 59.91 hours. In a practical clinical setting, this proves that the laboratory technicians must wait roughly 60 hours for the initial batch to multiply into the 1 million cells required for the patient's transplant. Mathematics directly guides the logistics of healing. We have covered a lot of ground today. Let us summarize the most important ideas. First, embryonic stem cells are the ultimate foundational building blocks, possessing the remarkable pluripotent ability to become any cell type in the body. Second, they are harvested from the inner cell mass of a microscopic blastocyst. Finally, Because they divide indefinitely, we can use exponential growth mathematics to precisely plan life-saving medical treatments. By mastering both the biological nature and the mathematical growth kinetics of these cells, scientists are unlocking revolutionary new ways to heal the human body. You now understand the science and math behind this medical frontier.